I really hope that the audio works this time. I'll check it before I post this video. All right, so this is the DNA structure and replication assignment. This PowerPoint is your quiz grade, which is one of the reasons why I wanted to post a video to help you out, because it's a quiz grade. So first, before I do anything, I want to point out this picture. We have DNA on one side, and then this picture over here is that photo 51 I was talking about that Rosalind Franklin took that helped to determine the structure, the helical, I should say twisted, ladder, double helix structure of DNA. And you can see some of the stuff labeled over here, the sugar phosphate backbone, where it's alternating sugar and phosphate groups. And then your nitrogen bases here in the center, where adenine and thymine go together, and guanine and cytosine match together. So let's get into this part. So this first one, you're just gonna drag and label. So click and drag these over to label. Make sure that if you're having trouble that you enable edit mode, because sometimes when you download things, it puts it into a safe mode, so you can't quite edit it, so make sure that you edit it. Also, I want to point out that pyrimidine and purine, remember, those are the types of nitrogen bases, so there's a little hint where they end up going, where your pyrimidine, it was your single ring, so it's going to be the smaller nitrogen base. That's your little hint. And the purine was the double ring, so it's going to be the larger nitrogen base. It's going to be twice as big as the pyrimidine. Deoxyribose is the sugar. Nucleotide, remember, nucleotides are your monomers for nucleic acids, and they consist of a phosphate, a sugar, and a nitrogen base. So there's another little hint for you on that. And if you need help, there's the Amoeba Sisters old DNA video I found to be very helpful for this. All right, so this one is actually a image to show DNA replication. So again, I've linked a video to help you out. I know there's a lot going on here, so I want to point out a couple of things. I'm going to start over here on the right-hand side. You have the 3' and the 5' side. Remember, that talks about how the DNA points, or the deoxyribose, the sugar, points, and they're opposite. So if you have 3' on this side and you follow this all the way down here, you should have 5' there. Hey, look, I gave you an answer. Just like if you follow this 5' right here all the way, you end up at 3'. So one side to the other, they end up opposite. This topoisomerase, this very large green circle, is an enzyme that helps to hold it apart while the helicase unzips. I'm giving you some hints here. Again, you're going to click and drag these to label the diagram. Use this video as much as you need to, double check it. Start with the ones I helped you out. I understand that some of these are enzymes. The ones that end in ASE are actually enzymes. Remember the difference between the leading and the lagging strand. The leading strand is continuously laid down, whereas the lagging strand is done in fragments. Remember, those are those Okazaki fragments that need to be put back together later by the ligase, the linking or the gluing enzyme. And then on your next slide, you have to type in these labels. So it's better that you do the one before it, so that when you come in here and you click on the box and you type in your answers, you can just use that, and it's just putting this into a different, a different format. I also linked the Crash Course DNA Structure and Replication video. That's the one we watched in class. So use that to help you out. I mean, if you want to go through and watch all the videos and then go do the assignment, that's fine. Or watch a video and do one slide. Watch a video and do one slide. It's really up to you. Make sure to save your work. There's nothing more frustrating than uploading something and realizing that it didn't save anything. And I'm emailing you going, hey, I can't grade this because there's nothing to grade. All right, next slide. I don't know why it won't let me go to the next one here. That is so weird. It did this last time. I know there's 2 more slides on here. There's one where you have to rearrange them in order for DNA replication. There's the steps. You have to actually click and move them. So the one that's on the top should be the first step, and the one that's on the bottom should be the very last step. So again, watch the videos to get some help. And then the very last slide is your identifying part. So use the information you put in the earlier slides to identify the parts, the different types of enzymes. Like the helicase is the one that does the unzipping. The topoisomerase is the one that holds it together so that it doesn't wind back up. If you have any additional questions or concerns, please let me know. Again, I can't stress enough, save your work, and make sure to upload it before the due date so I can get those grades in. Fingers crossed that my audio worked this time. I guess if you're watching this, then that means it did. All right, let's